I wanted to talk to you a little bit about the work that we're doing with JAK inhibitors, specifically with Baricitinib, and we're looking at it as it relates to HIV-associated neurocognitive impairment. So I won't belabor this point too much, since everyone here is certainly very familiar that a major barrier to HIV cure, as Steve Deeks really touched upon earlier, is a variety of mechanisms, but the persistent inflammation that is in individuals, even with well-controlled viremia, is thought to have major implications with respect to establishment, maintenance, and expansion of the HIV reservoir. And to that end, there's an unmet clinical need to be able to block that inflammation in a safe, specific, potent fashion. And that is really what we were coming after with respect to the JAK inhibitors. So our hypothesis going into this is that if you blocked some of these HIV-specific inflammatory events as it relates to a JAK inhibitor, what happens is you're downregulating phosphorylated STAT5s, phosphorylated STAT3. In turn, you have downregulation of a variety of factors, including downregulation of BCL2. You have reservoir persists, divides, expands, et cetera. But if you block all of that in the context of a JAK inhibitor, what happens is you have downregulation of the reservoir lifespan, which we've shown and I'll walk you through. You also have decreased homeostatic proliferation by blocking IL-7 and IL-15 driven reservoir expansion. You also block reservoir reactivation. So in doing so, the idea here in an ideal situation, which I'm going to walk you through what we've done so far, is that you could potentially have cessation of heart without viral rebound or delay in rebound by controlling the inflammatory events that degrade the viral reservoir over time. So what you see here is sort of the backdrop to what I'm going to walk you through, which is the idea that markers of reservoir persistence, reservoir size are correlated with elevated phosphorylated JAK-STAT pathway. So the idea here is that on the y-axis you can see the CD4 T cell counts, the elevated phosphostats, PD-1, persistence marker. These are all associated with elevated phosphorylated STAT pathway and size of the reservoir. So this really sets the stage for the idea that if we block JAK-STAT signaling in a safe, specific, potent fashion, that we could turn these markers back where we would like them to go, which is not to persist and not to have a large reservoir. So one of the first things that we did is that we looked at two of the JAK-STAT inhibitors with respect to T cell markers that are associated with persistence, increase in reservoir size, and increase in viral reseeding ex vivo. So these are in CD4 T cells from viremic donors. So you can see that we looked at proliferation on panel A, CD25, HLA-DR positive, 38 positives, and then also PD-1 being, of course, a marker for the reservoir size and persistence that's been shown eloquently by many others and probably some here as well. So we're showing in a dose-dependent manner that we can decrease these markers. That's a favorable thing. And so we wanted to be able to then take that and ask questions specifically about the reservoir itself. So what you see here on the next slide is that JAK inhibitors actually reduce the frequency of cells that are harboring integrated HIV DNA and CD4 T cells from HIV positive individuals. It's dose dependent. It happens without the presence of antiretrovirals. You can see here it's statistically significant, and most importantly, it is transpiring at clinically relevant concentrations that can be found during the steady state in humans receiving these agents, which are FDA approved. So then we wanted to ask a question about reservoir reseeding. Two microphones here. So reservoir reseeding as it relates to expansion coming out of the cells. So what we did is we took cells that are from HIV positive individuals. We then cultured them plus or minus ruxolitinib, and we did it at this time 1 micromolar. We've done this in a dose-dependent manner as well, and we've seen the same trends. And we asked whether or not the reactivated virus comes out, how much it comes out in the presence of the JAK inhibitor. So what you see here is the DMSO control, so not much comes out. You see that when you add the IL-15, plenty of virus comes out as expected. And in the presence of the JAK inhibitor in the right-hand side of each of these, we can see a significant reduction in the amount of virus that is able to reactivate in the presence of the JAK inhibitor. So we know this is being controlled through JAK-STAT signaling and IL-15. So that then set the stage for another question about viral reseeding as it relates to bystander cells. So we wanted to understand if we have a JAK inhibitor in place and there's a productively infected cell and then there's an uninfected neighbor cell, can we stop that neighbor cell from becoming infected? So what we did is we started with cells that were uninfected and we split them in half. Half of them, we added a cell tracer violet on them so we can see what becomes of them over time. And then the other half we infected with a GFP positive virus. And so then what we did is we mixed them together. And then what we did over time is we quantified the purple cells who were uninfected to see if they became green. So you can see on the right-hand side over here, what we're looking for are double positive green-purple cells, meaning that you were a bystander cell who then became infected from being next door to your neighbor. And so we're able to see in a dose-dependent manner statistically significant decrease in bystander cell infection, which we were pleased about. Again, downregulation because of the presence of the JAK inhibitor at clinically relevant concentrations. So then switching gears a little bit, we started asking questions as it may relate to the CNS reservoir, and in particular with respect to baricitinib, another JAK1, JAK2 inhibitor. So what you can see here are the drugs. So this is baricitinib, and I'm going to walk you through why we picked it on some of the next slides. But I really wanted to give you a bit of a flavor of what it can do up front in summarized form. So we looked at the antiviral potency in lymphocytes compared to 3TC in all cases. We also looked at it with respect to macrophages in both cases. Submicromolar and clinically relevant concentrations confer viral inhibition. And then we asked questions about viral reseeding and reactivation. So we looked at it in either primary T cells, so we were able to reduce the frequency of non-dividing P24 positive lymphocytes, and also the frequency with respect to reactivation in the JLAT system. And then we moved on to the reservoir size and macrophages, and then markers of activation in macrophages. So we looked at whether or not we could block reactivation in a primary macrophage model that we learned from Mario Stevenson's lab, and we were able to do that as well. We did that without toxicity and again at clinically relevant concentrations. So because of what we saw here in vitro, we wanted to then move forward to ask questions because of the effects in macrophages. What about HIV-associated neurocognitive dysfunction, which is largely driven by myeloid inflammation and infection within the CNS compartment? So I won't spend too much time on this. I know a lot of people are quite familiar. HIV-associated neurocognitive dysfunction is present in up to half of HIV-infected individuals, even those with periphery-controlled virus. And the idea is that there's a lot of trafficking, activation, inflammation that transpires in the periphery that then allows for translocation, infection and inflammation within the brain, both within microglia as well as bystander cells such as neurons undergoing neuronal dendritic simplifications and astrogliosis, et cetera. So what we wanted to look at is baricitinib being a JAK1, JAK2 inhibitor. It's potent anti-inflammatory. We know that a lot of these inflammatory events drive hand. Can we apply baricitinib to ask questions about hand in vivo? So before I go there and show you, I wanted to show you why we're kind of moving from ruxolitinib to baricitinib. So you can see ruxolitinib on the left-hand side, and this is its clinical profile. It was approved in 2011. It's BID dosing, it's orally bioavailable, that's great. It is hepatically cleared, which means that you do get some metabolites and potential CYP3A4 interactions with some other agents, potentially protease inhibitors. And again, it's BID dosing. So we started looking at baricitinib, which is once a day dosing. It's renally cleared, so it's very, very clean. What goes in, goes out. There's not metabolites. That has a much longer half-life, and in some regards it is more potent than ruxolitinib with respect to its in vivo dosing. It's only 2 or 4 mg once a day versus 10 to 25 BID for ruxolitinib. So for those reasons, I just wanted to give you a flavor of why we're looking at baricitinib and why we think it could be more favorable moving forward clinically. So we began to do some work with the JAK inhibitors, us as well as some others. Vicente Planellis' group looked at JAK inhibitors, and we recently published some of this work in PLOS Pathogens with respect to the reservoir. So it really set a stage for us to be able to then ask more detailed questions in vivo about this agent. So we went into this with a hypothesis that baricitinib can reverse hand in our murine hand model and potentially in humans. Due to the fact that it's a potent anti-inflammatory and its ability to block HIV replication, reseeding, and reactivation. We looked at that in both macrophages and microglia in vitro. So what we did is we used this SCID hand model where we start with a SCID mouse and we take human macrophages, we infect them in cell culture, and then once they're good and infected, we do a direct intracranial injection into the mouse brain. And then a number of days later, you come back and measure their ability to remember things by object recognition. And then at that time, once you've seen that they have neurocognitive impairments, they don't remember things, we give them the intervention of the JAK inhibitor in this area right here. And then we come back again and we ask about object recognition. We ask the mice to tell us, do you remember what you saw yesterday? Do you remember what you saw 2 hours ago? And see if the drug can help reverse it. And so after that's all done, then we harvest the brains. We do flow cytometry on them to look at a variety of markers for hand, P24, activated macrophages, astrogliosis, neuronal dendritic simplifications. So the first thing we wanted to ask before we did that, very simple question, does it cross the BBB? If it doesn't, donezo. And so we wanted to do that quickly, so we did. We looked at it in 5 different animals. We did traditional PK. And what you can see here is, I won't spend too much time, but there's linear pharmacokinetics here, which is fantastic. That's what we would hope to see. The drug goes to the brain, it stays there, it hits a steady state. Okay, so it goes, so now let's ask the more fun questions. So we wanted to understand if we can reverse now hand in the murine model. So this is this area here where the mice are confirmed to have behavioral abnormalities, and then we come in with our intervention. So what we do to test them is we put the mouse in here, and we show him 2 balls or 2 objects. I'm just showing red balls here. And we ask him to spend some time acclimating himself, remembering that he's seen these objects. And then we come back either after 5 minutes or after 2 hours, and we switch out one of the objects. And then we measure the amount of time he spends playing with the old object versus the new object. So that becomes a quantifiable discrimination index, where if he doesn't remember that the blue one is new, he's going to spend half his time here and half his time there, and he doesn't know the difference. But if he thinks, oh, that blue one's new, I remember that it's new, he'll spend more time there. So you can quantify that in terms of discrimination. So what you can see here on the y-axis is we tested 2 doses of baricitinib. They are meant to model the 2 mg and 4 mg once per day dose in vivo. We did that pharmacokinetically. You can see
Go ahead, keep going.
Okay. You can see on the y-axis, this is discrimination index, and then you can see that the uninfected controls remember very well, and then when they have HIV, they don't. And then when we give them the 2 concentrations of the drug, they are able to remember again. It reverses their behavioral abnormalities. And then we wanted to look at cellular markers to go along with that. So we looked at both doses again to block HIV infection as well as HIV-induced activation and inflammation. So you can see on the left-hand side, these are activated murine microglia, so MHC class 2 45 double positive cells. With the HIV infection, we see that go up, and with the doses of drug, we see it go back down. And then on the right-hand side, we also see HIV-infected P24 positive activated human macrophages. With HIV infection, that goes up, of course, and with the both doses of drug, it goes back down. And I'm just summarizing here. We also looked at astrogliosis and neuronal dendritic simplifications, which followed the same trend as the reduction in P24 and the behavioral abnormalities. We were able to show that HIV replication in primary human macrophages and microglial cells was blocked with this agent, that reactivation was blocked in vitro, and also activation with respect to monocytes. And we were able to couple that with our conclusions in vivo, which is that this drug crosses the BBB, is able to confer reversal in behavioral abnormalities, as well as reversal in hand markers phenotypically. So because of this data and the fact that it's FDA approved, the baricitinib is a treatment for hand that really needs to be evaluated in humans. It's a drug that's FDA approved. We know it crosses the BBB, and in mice, it completely reverses what we see in terms of behavior and phenotypic markers. So to that end, this is our team, Dr. Shanazi, of course, and then a number of us working together, as well as Vince Marconi, who's working on the human trial for ruxolitinib. So I'm happy to take any questions.